Hi, my name is Adeniyi Olagunju. I'm a pharmacologist based in the Department of Pharmacology and Therapeutics at the University of Liverpool. And I also say thank you to the organizers of the World Conference on Pharmacometrics 2022 for inviting me to be a part of this symposium. I'll be speaking about pharmacometrics in pregnancy, bridging the exposure safety gap. I consider this a very important subject, and I'm very eager to share my thoughts with you. I have no conflict of interest to disclose. In this 1959 advert, thalidomide was said to be free from untoward side effects. A few years later, it became very clear that in utero exposure to thalidomide was responsible for embryo-fetal development toxicity. The little girl that you see here is Tracy Benham. Her limb development was affected by in utero exposure to thalidomide. These are the prosthetic limbs that she used throughout her childhood and teenage years. There were thousands of other kids like her. Preventing this type of tragedy without denying pregnant women access to clinical trials of potentially life-saving medications, we need a paradigm shift in the assessment of embryo-fetal development toxicity during clinical drug development. These are the things that I'm going to be sharing with you. 90% of women have to take at least one drug during gestation for any of these or other conditions, which may be obstetric specific indications or chronic conditions or even acute conditions, often without adequate supporting evidence from clinical trials. For women who live with any of these and other conditions, the time of pregnancy often becomes time to choose between the real pain of living with these conditions or the unknown risk to their baby if they take a medication. Added to this is the potential that the physiological changes associated with pregnancy may alter drug exposure, which is a key determinant of efficacy and safety. For instance, we know the impact of pregnancy on certain cytochrome P450 enzymes. Their abundance and the activity often change due to hormonal influence on their transcriptional regulators. Over the years, published studies from several pharmacology groups have evaluated what this means for several drugs. Those on this slide are some examples from our group. In a series of papers published between 2014 and 2 years ago, we show that pharmacogenetic variability may worsen pregnancy-induced changes for certain drugs. We have also provided evidence that drug-drug interactions worsens pregnancy-induced changes in drug exposure. Several key developments in the last few decades have resulted in significant improvement in our ability to take all of this information and integrate them within physiologically based pharmacokinetic modeling framework, where we combine key system parameters with drug-specific parameters in order to give realistic predictions of drug administration, absorption, distribution, metabolism, and elimination. And what we come up with is a multi-compartmental model like the one presented here. This workflow illustrates how a maternofetal PBPK model could be derived from a non-pregnant adult model by stepwise introduction of key changes. In this 2012 paper, the authors published a database of equations for modeling gestational age-dependent changes in key parameters. This is an excellent example of data sharing and collaboration, which has facilitated further development in recent times. The list of available equations for modeling these changes has significantly expanded over the years as more groups publish equations on previously unexplored areas. One of the other key considerations in modeling prenatal drug exposure is the difference between an adult circulatory system and a fetal circulatory system. This image of fetal circulatory system shows the presence of two right to left shunts, which are small passages that bypass certain body parts, in particular the lungs and liver, which are not fully developed in utero. In our maternofetal PBPK model, we account for these differences, leveraging published clinical data. Several maternofetal PBPK models have been published over the years, especially in the last 5 to 6 years. In this example, the authors first used in vitro studies in transwell to determine the efflux ratio of drugs that are substrate for the human efflux transporter, PGP. Then, using proteomics-informed efflux ratio relative expression factor approach, they predicted the fetal unbounded UC ratio for these drugs at term. This allowed for the extrapolation of in vitro transfer data to in vivo placenta data, and it was implemented within their PBPK model. The integration of the active transport model in this approach used by the author, this integration of active transport significantly improved model predictions, as we can see here. The second example I want to share with you is a maternofetal PBPK model, which we use within our group to characterize prenatal drug exposure across gestation and to model organ level fetal drug exposure. In this study, we compared thalidomide with evavirenz. What is striking here is the significantly higher accumulation of thalidomide in fetal circulation compared with evavirenz. We also explored the concentration of both drugs in fetal brain. Using the AUC ratio of the drug in fetal brain to maternal plasma, the model predicted thalidomide exposure to be more than 4 times the maternal drug concentration. For evavirenz, fetal brain exposure was below 10% of maternal plasma exposure. And I want to call your attention to the following posters that are available at this conference where maternofetal PBPK models are reported. No matter how comprehensive these models might be, they have their own limitations. And here are 2 of them. Placenta modeling is limited by the quality and the quantity of available data. And unfortunately, these are still very limited at the moment. Secondly, exposure characterization does not give us answers to questions about safety. In other words, maternofetal PBPK models are unable to answer the and so what question. For instance, HIV nucleoside reverse transcriptase inhibitors are preferentially distributed to fetal compartment, and fetal to maternal ratios of up to 4.0 have been reported, but these drugs, most of them are not associated with any major safety issues. During preclinical drug development, embryo-fetal development toxicity is evaluated in 2 species, usually rat and rabbit. These animals are exposed to study drug during organogenesis, and these endpoints are then assessed. Multiple of exposure index, MOE, is obtained by comparing the no observable adverse event level in animals to human therapeutic dose. What are the problems with this? Placenta is the most important organ in embryo-fetal development toxicity, but there are big interspecies differences in placenta structure and function between animals and humans. And there are also massive differences even at different developmental stages. Secondly, embryo-fetal development toxicity studies are observational in nature. They assess primarily morphological abnormalities. They are not designed to evaluate key events that are important for embryo-fetal development, including neurodevelopment, immune system development, or the effect of drugs on the endocrine system. In some cases, animal models may be able to mimic some human phenotype, but then the underlying mechanisms are often very different. And what this implies is that this has resulted into is drugs that are developed based on insight from animal models do not always translate well when they are progressed to clinical trials. For instance, of 12% of drugs that successfully advance beyond preclinical stage, less than 12% go on to make it to the market. And as you can see here, hepatotoxicity is the single most common reason for this attrition. The clinical trial success rate can theoretically be improved to 56% if failures that are due to toxicity are prevented. And as you can imagine, pregnant women are even completely missing in this picture. And what this means is it results in a lot of uncertainties about the safety of drug in pregnancy. And so women are excluded, pregnant women are excluded in clinical trials. And paradoxically, it leads to widespread off-label use of drugs without any supporting or safety evidence in pregnancy. So clearly, we need a paradigm shift in the way we assess the safety of drug during pregnancy if we're going to overcome the current inertia in pregnant women inclusion in clinical trials. Over the last 5 to 7 years, advances in tissue and biomedical engineering have facilitated significant progress in vitro models. Although static 2D cell culture models are still widely used, now we have 3D culture models. To illustrate, consider organ-on-chip as an example. Organ-on-chip models are advanced miniaturized functional models of organs constructed from multiple cell types that are co-cultured within microfluidic devices like the ones shown here. They are designed to recapitulate the functional unit of substructures of a given organ. They are also capable of incorporating organ-specific features and environmental cues that are important for cell and organ development, including the three-dimensional structure of fluid flow as well as stretch. They enable modeling of organ function and physiology or pathophysiology responses in real time. Reported, there are several of them that have been reported, but here we're going to focus on the organ-on-chip model. This three-dimensional cross-section of the human placenta illustrate what we're trying to achieve. So here we see the cotyledon, which contains chorionic villi that is bathed in maternal blood, which is located in the intervillous space. A zoomed-in view highlights the fetal capillaries that are located within the chorionic villi. The placental barrier that separates the maternal intervillous space from the fetal capillary lumen consists of two cell types, the syncytial trophoblast and the fetal endothelial cells, separated by a thin interstitial tissue layer. Several model systems, in vitro model systems, have been used over the years to try to recapitulate this. The placenta-on-a-chip model has arisen in the last couple of years, precisely between 2016 and 2018. And I would like to share two examples with you. In this particular study, the placenta-on-chip model was shown to recapitulate key aspects of placenta physiology and function, including drug transport. Widespread expression of the placenta efflux transporter, BCRP, was confirmed on the maternal side, and its role in limiting substrate transport to the fetal side was demonstrated. In this second example, the placenta-on-chip was exposed to titanium dioxide, and that led to dose-dependent production of reactive oxygen species. The resulting loss of tight junction proteins led to perturbations in the placental barrier permeability, even at very low concentrations of titanium dioxide. And what this means is the placenta-on-chip model is capable of recapitulating both the placenta function that we observe in vivo, as well as the toxicity that may result from exposure to toxic substances. It is not just placenta-on-chip model. Models for other organs, like I highlighted earlier, have also been reported. In this example, this study, the liver-on-a-chip model, was published. And this model recapitulates the morphology and the functional features of the human liver. In fact, it correctly identified 27 drugs that passed animal testing, but later caused hepatotoxicity in humans, with a sensitivity of 87%. In fact, it is predicted to be able to generate $3 billion annually due to increased R&D productivity if this is widely adopted within the pharmaceutical industry. Now, how do we make use of all of this development? In a report of the National Council in the United States, convergence of the life sciences with other fields is identified as the key strategy to tackle complex challenges and achieve new and innovative solutions. Importantly, organs on chips were identified as an example of such convergence. This requires transdisciplinarity, which transcends disciplinary approaches to research. It requires that we begin to view the different components that are required to implement this kind of approach as tools, not as disparate disciplines that have nothing in common. Leveraging developments in both PBPK and organ-on-chip modeling to create this type of convergence requires development in 4 key areas. First is openness in pregnancy PK data sharing to improve qualification of more maternofetal PBPK models, which can be used to generate granular maternal and fetal exposure predictions. Placental on a chip model informed by clinically relevant exposure predictions from PBPK models. This can be used to characterize drug metabolism and transport, which can enable further refinement of PBPK models. Importantly, it will capable of capturing drug-induced perturbations in placenta morphology, proteome, and transcriptome. The third component in this approach is a group of organs on chip for characterizing drug-induced perturbations in developing fetal organs. And the fourth component is bioinformatics analysis of proteomics and transcriptomics data, which may lead to discovery of molecular signatures of drug-induced perturbations that can form the basis for risk assessment conclusions. The goal is to develop an integrated platform like this, which can facilitate an understanding of which biological pathways are enriched in differentially expressed genes and proteins. This can progress to the development of adverse outcome pathways, where molecular key events that result from prenatal drug exposure can be used as part of the toolbox to predict which drugs are more likely to result in adverse outcomes and which one can safely be advanced to clinical trial in pregnant women. The question is, will this really happen? Or put in another way, is this achievable? But the real question is, do we want to be left behind? Next generation risk assessment in the cosmetic industry already involve a high-level integration of different tools. PBPK modeling is used to generate measures of systemic exposures resulting from topical application of products. The predicted levels of exposure is used to inform bioactivity experiments to discover differentially expressed genes resulting from drug exposure and cellular stress pathways. Decisions about margins of safety are then made using this integrated knowledge, all of this without animal experimentation. And of course, a major requirement for regulatory acceptance of this type of model that we're proposing is defining the context of use. The context of use, in the words of the FDA, is the manner and purpose of use for a drug development tool. It is qualified for a specific context of use. The context of use statement should characterize all of the elements characterizing this purpose and manner of use. The qualified context of use defines the boundaries within which the available data adequately qualifies the use of the drug development tool. As additional data become available from further studies, the context of use for any particular tool can then be expanded. So far, I've emphasized embryo-fetal development toxicity, but Fertility and early embryonic development, as well as pre and postnatal development, are also very important. And it is clear that because of the vast differences in developmental stages involved, what is suitable for embryo-fetal development toxicity may not be suitable for these other ones. Hence, what is required is the development of a battery of tools for each application, and each one of them qualified for a specific context of use. To summarize, key development for next generation reproductive and developmental toxicity assessment are already available. Likely desirable attributes will include the fact that it needs to integrate clinically relevant model-informed exposure metrics integrated with human relevant in vitro models, each one adequately qualified for its context of use. The cost may be high, but the gain will be a lot more. Humanizing preclinical reproductive and developmental toxicity assessment is the only way to translate achievement in maternofetal PBPK modeling into measures of safety. This is crucial for progress in evaluating drug safety during pregnancy. We conclude by thanking you for your patience, and I want to acknowledge my colleagues at the University of Liverpool, my former colleagues and collaborators at Obafemi Awolowo University in Nigeria, as well as colleagues in other institutions, as well as Wellcome Trust for previous funding. Thank you for your attention.